Okay, so we'll be continuing with the glycolysis. And today, what we are going to talk about is that you have already learned all the steps in glycolysis, and you have also um learned that instead of glucose, you can have other source of energy. For example, you can have fructose, you can have lactose or galactose. So we'll be talking about if some problem occurs in the metabolic activity, what happens? Do you have some, disorder or something happens during this period? And third thing which we are going to discuss today is that how this glycolytic pathway is being controlled. So what are the factors by which either there will be increase in glycolytic pathway, or there will be inhibition of glycolytic pathway. So our purpose would be to understand the regulation of glycolytic pathway. Now, one thing which I would like to clarify here is that whenever this reaction occurs from glucose to pyruvate, you find that there are certain stages where energy is involved. Energy is involved means either ATP is being consumed, for example, conversion of glucose to glucose 6 phosphate, or for example, fructose 6 phosphate to fructose 1,6 bisphosphate. These two places, the ATP is being consumed. That you already know. Then there are two steps where the ATP is being synthesized, and one of them is here phosphoglycerate kinase, and second one is pyruvate kinase. So there are four steps where the energy is involved, and you will see that these steps, they are irreversible steps. So this step is irreversible, whereas glucose 6 phosphate to fructose 6 phosphate, it is a reversible reaction. So what I mean to say here is that the control of metabolic activity you will find is happening at those places where the reactions are irreversible. Why it is so? Because the energy is involved, and suppose you require an energy for the reaction, as per law of different laws, you cannot utilize the same amount of energy for reversing the reaction, because some of the energy is being lost, because any process cannot be 100% efficient. And that is why you find that the steps which are being regulated are the one where energy is involved. So these points you keep in mind, and we will discuss about this. We have already discussed that one of the way of controlling it. Can you see these steps? Let me just point it out. So there is one step where you have uh, this is the step. Can you see this step where NAD is involved? So what happens if NAD plus, which is being utilized, if it becomes limiting all the steps of this reaction, that stops. So you have learned how this NAD can be recycled. Can you tell me how this NAD is being recycled? Any one of you? We have discussed in the last class. This is very important steps where NAD plus recycling is important. So that part we have discussed. Let me just give you again this idea. What happens when pyruvate, till pyruvate synthesis, NAD is being utilized. But there are three ways in which this NAD can be regenerated. One of the way could be ethanol fermentation, where pyruvate is converted into acetaldehyde, and this acetaldehyde is converted into ethanol. And during this process, NADH, which was generated on this steps, this is the step where NADH was formed. Sorry, this is the step where you have seen formation of NADH. This is the step where NADH is formed. Now this NADH is, unless it is consumed, you don't have NAD plus in the cell, and then the reaction will stop. So what you require is, you require essentially regeneration of NADH. And this is the step where you see the regeneration of NAD plus from NADH, which was formed in the step I have just shown you. So this is one way. Second process you have learned is not only by formation of ethanol, you can also do the same kind of NAD plus regeneration by formation of lactate. So there's the second way by which you can regenerate NAD plus. So this is basically maintaining the redox balance. And this redox balance is responsible for continuation of glycolytic pathway. Now these two cycles, they are happening when or these two reactions are happening when there is an anaerobic condition, when the cell is having no oxygen or very little oxygen. So under those conditions, under anaerobic condition, you have seen either ethanolic fermentation or lactic acid fermentation occurs. On the other hand, NADH generation is very important because, as you have seen, this NAD plus which has been formed, they should be regenerated, and that can happen when the acetyl CoA is formed. How acetyl CoA is formed? This is the reaction where you have pyruvate NAD plus coenzyme A, it forms acetyl CoA. Of course, here NADH is formed, but this is not the step which is a regeneration. What you have is that NADH is being consumed when there is an aerobic condition. Under aerobic condition, NADH is being consumed in oxidative phosphorylation. This part of oxidative phosphorylation we will discuss later. And let me tell you that oxidative phosphorylation is a major source of energy, major source where your ATP is being produced. I will go back again to show you that in this glycolytic cycle, what you have seen is that at the end, 2 ATP is formed and 2 molecules of NADH is being generated from NAD. So these 2 NADH will be reoxidized to NAD plus by oxidative phosphorylation. I'm not giving you the steps of oxidative phosphorylation. We'll discuss that as a separate chapter. So what happens? The electrons from NADH will move to molecular oxygen because oxygen is present, and that is where the electron transport cycle will happen. The electrons will be, moving from NADH to molecular oxygen. And during this process, the protons will be pumped out of the mitochondria. And as you know, that in eukaryotes, mostly it is an aerobic condition. And under aerobic condition, when protons are pumped out, the proton will re-enter. And during that process, ATP is synthesized. We will talk about ATP synthesis separately. So there's a very complicated mechanism where the ATP is generated from NADH. Here, what you have seen is that ATP is generated by in case of pyruvate kinase, what has happened? ADP and PI mixes and makes 2 ATP. And here also 1,3-phosphoglyceric acid to one of the phosphate group gets attached with ADP. What is happening? These molecules, they are energy rich molecules. If you look at their delta G or their structure, they are structurally having high phosphoryl group transfer potential, and because they have high phosphoryl group transfer potential, they can transfer their phosphoryl group to ADP, and that's why they form ATP. And these reactions where ATP is synthesized because of high phosphoryl group transfer potential of substrate molecule are known as a substrate level phosphorylation. So this part also have given you idea about this. So you have learned so far that maintenance of redox balance is one way by which glycolysis can be controlled. You have also learned that the glycolysis have two steps where substrate level phosphorylation occurs. You have also learned that there is possibility of regeneration of NAD plus by ethanol fermentation or lactic acid fermentation. And third one, which we will discuss in greater detail by oxidative phosphorylation. Then we have also talked about how dehydrogenases work, and we have also talked about how fructose and galactose, they are being metabolized. Today, we are going to further probe into the galactose metabolism. Suppose there is lack of galactose metabolism, or suppose there is a lack of conversion of lactose to galactose, there are problems, and that is what we are going to discuss about. So here, just to recapitulate, what you have done is that galactose, this galactose is formed from where? What is the molecule which forms galactose? It is a lactose which is converted into glucose and galactose in presence of enzyme lactase. That part also we will discuss. But here what you see is that galactose is converted into uh, galactose 1-phosphate, and here this galactose 1-phosphate, UDP glucose, these two, what will happen, that galactose 1-phosphate can be converted into glucose 1-phosphate. So this is basically what you are doing, is that converting galactose to glucose 1-phosphate. How? First step is conversion to galactose 1-phosphate, and this galactose 1-phosphate in presence of a transferase, and this very important enzyme. Remember, I will just talk about how, if this is genetically absent or it is absent in the cell, what could be the problem. So what you have learned is that galactose can be metabolized by galactokinase. Then second is galactose 1-phosphate uridyl transferase, which will convert galactose 1-phosphate to glucose 1-phosphate. And then this UDP glucose, can you see this UDP glucose, which is important for continuation of reaction? So what will happen? Once it is converted into UDP galactose, how the cycle will be converted? So what will happen, that this UDP galactose is converted into UDP glucose, and that will be recycled. So it is coming back over here, and then it will be again utilized. So actually, what you have learned here is that even if galactose is not present in the system, still galactose can be synthesized. Can you tell me how galactose can be synthesized even if it is not present in the system? Tell me, look at this reaction and tell me how we can synthesize galactose. Even if galactose is, suppose a doctor says don't take milk, and in that case, that means you don't have any source of galactose. From where galactose will come? Looking at this reaction, can you tell me? Okay, can you tell me, can you think about your previous classes and tell me why galactose is important? Is there any important reaction where you have, seen galactose is very important? Any one of you? You have learned that the galactose is required for the recognition of sialoglycoproteins. When it becomes a sialoglycoprotein, galactose is the terminal point. So the synthesis of sialoglycoprotein is very essential, because there are many proteins which will be working because of the presence of galactose. So what I mean to say here is that even if a doctor says, okay, don't take lactose, they will block the, nothing, there is not a big deal, you just don't take milk or don't take source of lactose, what will happen? There is no generation of galactose, and that's the way things will happen. But you still require galactose. So from where galactose comes? Galactose comes from a reverse reaction where you have UDP glucose, and this UDP glucose in presence of epimerase can be converted into UDP galactose. So there are two ways in which this UDP galactose can be formed. Either UDP galactose can be formed from the glucose, or even it can be formed from galactose or from glucose. If it is from glucose, it is enzyme epimerase is important. If it is from galactose, it is this transferase is important. Now, this transferase is absent in one of the genetic disorder, which I'm going to discuss, and that's why I spent so much of time in explaining you this steps of reactions. Okay, this is already known to you about the fructose metabolism. Yeah, so what happens? There is a lactose which is present in the milk as a source of carbohydrate. It is one of the carbohydrate which is present, and so people, they take it. It's one which is listed as a very useful disaccharide which is present in the milk. What happens to this lactose? There is an enzyme called as a lactase. It will break this bond, beta glycosidic bond, and then you will have one molecule of galactose, one molecule of glucose. Glucose will be metabolized as such. Galactose, as I have shown you, that galactose will be converted into glucose 1-phosphate, and glucose 1-phosphate will be converted into glucose 6-phosphate, and that's the metabolism occurs. They will enter in the same glycolytic pathway. But what happens? There are people who have, deficiency of this enzyme. So if the deficiency of lactase happens, what will happen? There will be no metabolism, and the lactose will accumulate. Now, this lactose is the one which is very favorable for the bacteria. So this lactose, if it is lactase is not metabolizing lactose, so what will happen? Some of the microorganisms present in our gut or colon, what they do is that they ferment this lactose. And once they ferment it, lactose is converted into lactic acid, and also it generates small amount of methane and hydrogen. So what will do? These gas produce, they create uncomfort and flatulence. And that's why you will see that those who have lack of this lactase enzyme, they have problem in metabolizing lactose, and they have problem in their stomach or gut, and that causes flatulence. And this is because of the lack of this enzyme. So this enzyme is not there. If it is absent, lactase is absent, this will accumulate and the problem occurs. Now there is another problem which happens when galactose accumulates, because galactose becomes highly toxic for the cell. And let us see what are the toxicity which can happen because of the galactose. And when does it happen? You will see that the galactose metabolism is absent in case of galactosemia. Galactosemia refers that you have a deficiency of an enzyme, galactose 1-phosphate uridyl transferase. Just I will show you what this enzyme does. Just now we have discussed it. So this is the enzyme, which is this enzyme is deficient. This is not present. If it is not present, what will happen? There will be, galactose, which is not going to be metabolized. And if it is not metabolized, then what will happen? Let's talk about that. So under the case where the enzyme galactose 1-phosphate uridyl transferase, this enzyme, if this enzyme is absent, and which is a genetic disorder called as galactosemia, this is a galactosemia, and that is what leads to the accumulation of galactose. So what will happen? In many infants, in kids, this will lead to diarrhea. Once they consume milk, they will face diarrhea. There is, if it is not metabolized for a longer time, you will have a jaundice, and sometimes progressing of liver cirrhosis. Why? Because your liver, that has a lot of load, and it is not going to be metabolized. Another problem comes if galactose is present for a very long time in the body. What it does is that, especially in your eye, so in case of your absence of the activity in eye, what will happen? That galactose will accumulate, and this galactose is converted into galactose. So this is the reaction where aldose reductase. So this galactose is nothing but a aldose. Why? Because the aldehyde group is present. So this aldose is converted into CH2OH. Now this conversion from aldehyde to alcohol leads to the accumulation of galactose. It is the same thing like you have learned about the glucitol. Glucose can be converted into glucitol, galactose can be converted into galactitol. And what it does is that it leads to the formation of a cataract. Why? Because if it accumulates in the eye, galactitol, it leads to the cataract formation. Can you tell me that does it happen in any other disorder? Answer is the people, those who are suffering from diabetes, very high level of diabetes, they also have a cataract formation. What happens there? This glucose molecule, which is not being metabolized by the diabetic patients, that is converted into glucitol, and that is what leads to the cataract. So all these things are, metabolic disorders. So one must understand what happens. Now we are going to move on. Once you have learned about the metabolic pathway, where the entry of galactose in the pathway is very essential. It should be metabolized. It should not be accumulated. Once it accumulates, there is a problem. And as I said, if galactose is causing so much of problem, if doctors, they diagnose the absence of enzyme, what they say is that, okay, don't take milk. And you know that there is a small population which is, intolerant to milk. Now we have to discuss about how the glycolytic pathways are being controlled. I'm discussing this glycolytic pathway in detail because you will be learning that most of the metabolic pathway, they are being regulated, and their regulation is more or less in the same principles. So because we are going to discuss about the principle, you will understand the metabolic control. So here we are going to talk about how glycolytic pathway is being controlled. And let me tell you that the cell is very smart. It doesn't waste any energy. And what it does is that when there is energy required, then only you will see that the glycolytic pathway is moving towards the forward direction. Otherwise, the glycolytic pathway will be stopped. So you will be learning a tight control in glycolytic pathway. On the other hand, if you require a lot of energy, glycolytic pathway will move faster. If you are sleeping, glycolytic pathway will not be required, so they will be controlled. So how they should be controlled? They should be controlled on the basis of or on the demand of ATP. So ATP is the general currency, and that's why ATP is the one which is controlling the reaction of glycolytic pathway. So the production of ATP generated by degradation of glucose. So what is the role of glycolytic pathway? One thing is that it generates ATP. You have seen there is a very quick 2 ATP is being produced when glucose is converted into pyruvate. Second major thing is that whatever is being synthesized, they will be making building blocks. So one of the building blocks can be made by formation of, for example, fatty acid. It is not only fatty acid, it can also go into other pathways. So that part we will discuss at a later stage when we talk about the citric acid cycle or TCA cycle. But yes, as you know, any goal of metabolism is to generate energy and to synthesize different building blocks like nucleic acid, like your amino acid. Through amino acid, you can synthesize proteins, and fatty acid, and through fatty acid, you can synthesize lipids. So we'll talk about how these building blocks can be synthesized. So it's a very strange that on one hand, we are talking about the breakdown. Of course, glycolytic pathway individually cannot synthesize these things, but they will provide feed for synthesis of these molecules at the later stage. So in metabolic pathways, enzyme catalyzing essential irreversible reactions are potential site of control. This part I have already told you that in the cycle, the enzymes, something like, as I have already discussed, hexokinase. What hexokinase does? I think by so many times I have repeated. So you must be knowing what hexokinase does. Hexokinase takes any hexose, could be glucose, fructose, or other molecule, which can be converted into glucose 6-phosphate or fructose 6-phosphate. Okay, so hexokinase role is to convert the sugar molecule into their phosphorylated derivative. What is the role of phosphofructokinase? Phosphofructokinase, you have seen, is converting fructose 6-phosphate into fructose 1,6-bisphosphate. You will be learning that this is the enzyme which is very, very important, and it is the one which will be controlling most of the reaction. So you will be, we will talk about how this is involved in different control mechanisms for phosphofructokinase. The third one is pyruvate kinase. So all these are virtually irreversible reaction, and hence these enzymes are the one which are regulatory enzyme. Of course, they are providing catalysis because hexokinase will convert hexose into hexose 6-phosphate, like glucose to glucose 6-phosphate. Phosphofructokinase is converting fructose 6-phosphate to fructose 1,6-bisphosphate. But they are also controlling the rate of reaction. And sometimes those reactions which are being controlled by these enzymes, these enzymes may be known as pacemaker enzyme. They decide the pace of the metabolic pathway. Now, question is how these enzymes are being regulated. When we were discussing about the enzymes, we have discussed that the enzymes can be regulated by different mechanisms. One of the mechanism, which is a common mechanism, is by allosteric effector. Can you recall what is allosteric effectors? Can you recall what are allosteric enzymes? Any one of you? Allosteric enzymes are those enzymes where other than active site you have allosteric site. And what happens these allosteric site? These allosteric site are the one which are controlling the activity of enzyme. They will regulate the active site in such a way that it becomes functional or non-functional. It becomes active or it becomes inactive. So there is one way by which an enzyme can regulate a reaction is by allosteric effector. And most of the time you see that these allosteric effectors are a small molecule like ATP, like AMP. So they can regulate the enzymatic activity. They can enhance or they can lower the enzymatic activity, and that's the way they can control. Second thing is by covalent modification. What is a covalent modification? Here, the enzyme is not binding through non-covalent interaction, and there is no specific site where they can bind. It is not the allosteric site, but the enzyme is modified covalently, and this covalent modification could be phosphorylation reaction. So suppose you have enzyme, its serine group will be phosphorylated, and this has got modified. So this kind of a modification is known as a covalent modification. And when covalent modification occurs, the enzyme changes its structure. And when it changes its structure, what will happen that the change structure could be active or could be inactive depending on the requirement. And third way of regulating enzyme is by regulation of transcription. Now this is a different mechanism. So can you recall how transcription can be regulated? I have not discussed the detailed regulation of transcription, but yes, what is the transcription? Conversion of the gene or the DNA into RNA. And now, if I can control the RNA, which is ultimately going to synthesize an enzyme, which is basically a protein, then you have regulated the enzymatic reaction. So there are three levels of control. One is allosteric effector, where non-covalent binding or just binding at the allosteric site is the one which is regulating the enzymatic reaction. Second is a covalent modification by which the enzymatic reaction is regulated. Then third one is by transcription. Now, let us see how these things are different and when they are required, when they can be affecting the rate of reaction. These are being affected by the requirement of the cell. Do we require them quickly? We require them in few minutes, few hours, and so on? So what you see is that most fast reaction is allosteric effector. These reactions, they are occurring in millisecond. So your ATP is high or low, that will signal, that will bind, and then the enzyme will become active or inactive, and that reaction is controlled. Second is by covalent modification. That takes few seconds. First one is a millisecond. Second one is this reaction takes few seconds. And that one transcription, that is more at the gross level, and that requires time, and it can happen in hours. Because here, what you are controlling is you are controlling the conversion of DNA to RNA. Then RNA, if there is a less amount of RNA, then lesser amount of enzyme is being synthesized, and that's the way the control is happening. So that is a relatively lengthy process, and that control occurs. But yes, the most energy is going to be saved by transcriptional regulation, because ultimately you are not synthesizing protein. On the other hand, the most conserved or most fast reaction could be where already enzyme has been synthesized, you are just controlling its activity. Okay, so let us move on to the next way how these are being controlled. So we are going to talk about one of the enzyme which is very well known. Of course, we will be talking about other enzymes as well. So one of them is phosphofructokinase. Now recall what phosphofructokinase is doing in the glycolysis. Phosphofructokinase is the one which is converting fructose 1,6-phosphate to fructose 1,6-bisphosphate. And this enzyme is very important because it controls the mammalian glycolytic pathway. And many times this has also been referred as the pacemaker enzyme. This is the one, once this reaction occurs, once fructose 1,6-bisphosphate is formed, the enzyme becomes, or the product becomes committed for glycolysis. Because next step is breakdown in the presence of aldolase, where you get 2,3 carbon molecule dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So high level of ATP allosterically inhibits the enzyme in the liver. Try to understand. When there is a high level of ATP, that means energy is more. So what should happen? More of fructose 1,6-bisphosphate should not be formed, because already you have energy. You don't require energy. And remember, till fructose 1,6-bisphosphate, no energy is formed. Up to this step, only energy is being consumed. I will go back to the first slide and try to show it to you. Yeah, here what you find is that this is the one which I'm talking about. I'm talking about this enzyme, phosphofructokinase. This enzyme, once it is converting to fructose 1,6-bisphosphate, the reaction becomes committed. So this phosphofructokinase is the one which is a tightly controlled. Its reaction is going to be, its pace, its activity is being controlled by different steps, and it's very important to understand. And once it is being controlled, that means no energy is being produced, because most of the energy is produced in this part. The greener part is the one where energy is being consumed, and the energy is being produced in this part. So you will see that once we have controlled this enzyme, we have inhibited this enzyme, more and more ATP will not be formed. So let's go ahead and try to understand the mechanism. So you have this enzyme phosphofructokinase, which is a key enzyme for controlling the glycolysis. And what happens here is that high level of ATP, when you have high level of ATP, allosterically it inhibits enzyme in liver. So most of the synthesis is occurring in the liver. So what happens is that it inhibits the enzyme in the liver and lowering its affinity for fructose 6-phosphate. So the question is how it inhibits. What it does, it lowers the affinity for fructose 6-phosphate. That means what will happen to the Km? If affinity is low, that means
Km is high.
high. So what it will do is that it modulates the enzyme in such a way that Km becomes high, and that is the way it regulates. I will show you the reaction. So, and what happens the other one? What is the AMP? Can you recall what is the AMP? AMP is the one which is formed from ATP. So ATP can be converted into AMP, and that's the way the complete phosphoryl group ATP is being transferred. So AMP reverses the inhibitory reaction of ATP. So what is happening? It's very interesting. Phosphofructokinase is inhibited by ATP. That means when we have more energy, it is going to be inhibited. But on the other hand, so what is happening? ATP is the one which is inhibiting. So you will find that the ATP is the one which is inhibiting, whereas AMP is reversing the inhibition. So if I have ATP, that means if there is a lot of ATP, what will happen? Phosphofructokinase will be inhibited. It makes sense? When there is a lot of AMP, that means energy has been utilized, whatever the ATP reserve was there is over, so AMP will reverse the inhibition of ATP. What it means is that it is going to activate fructose phosphofructokinase. It will become active. So the activity of phosphofructokinase, you can see, is being regulated by the energy status of the cell. More energy is present, that means it will be inhibited. Less energy is present, that means its inhibition is being removed. That means, so the activity of enzyme increases when ATP is to AMP ratio is lowered. That means more of ATP is being consumed. In other words, glycolysis is stimulated as energy charge falls. And that makes a sense because you need to synthesize more and more ATP under those conditions. Now let us see another condition. Suppose the cell pH falls. So if pH is low, what it means? That there is a more pyruvate or there is a more protons. So more of the proton, they will prevent excessive formation for lactic acid. And because if there is a lot of lactic, from where this lactic acid comes? Lactic acid comes from the conversion of pyruvate to lactate. So if you're running, you are producing, you are not having enough of oxygen. So your pyruvate will not go into acetyl-CoA and the TCA cycle, and that will be accumulated. And once a pyruvate accumulates, it will be converted into lactic acid temporarily. And if lactic acid is more, what will happen? pH of blood drops. And let me tell you, even one pH drop in blood can cause acidosis. And that is what is avoided. So what it will do is that this HIN prevents excessive formation of this lactic acid, and that's why it inhibits phosphofructokinase. Now tell me how this inhibition of phosphofructokinase will inhibit lactic acid formation. If I inhibit phosphofructokinase, what it means? No further metabolism. That means fructose 1,6-bisphosphate will remain there. That means no pyruvate is formed. If pyruvate is not formed, lactic acid cannot be formed. Okay, so that's the way it controls the it controls the acidosis in the cell. Now, in some cases, what happens if you are requiring ATP quickly? What happens that ATP being rapidly utilized under those conditions, there is an enzyme called as adenylate kinase. Now, this enzyme, what it does is that it takes one molecule of ATP, one molecule of ADP. So you have one ADP, sorry, you take two molecules of ADP, and what you synthesize is one of the phosphoryl group from ADP is being transferred to from one ADP to another ADP, and you synthesize ATP and AMP. So this way, whatever energy charge is present is made in the utilizable form. But this reaction is not occurring so fast unless there is a high depletion of ATP and your ADP is in excess. So what I mean to say here is, if you recall when we were talking about introduction to metabolism, I have talked about that the ATP or adenylate pool is important. So what you look at is that you look at the energy status of the cell by concentration of ATP, concentration of ADP, concentration of AMP by concentration of ATP and half ADP. So that's the, sorry, concentration of ATP and half ADP and concentration of ATP, ADP and AMP. That is what is going to determine what is going to be the energy charge. And that you can refer to our introduction to metabolism where we have talked about this. So what happens in a short run? What happens that the ratio is going to be maintained. It's more or less in the range of maximum. It could be 1. Mostly it is maintained to 0.9. So in a cell, this pool is a constant over a short term. And second, that the concentration of ATP is greater than ADP and concentration of ADP is greater than AMP. So what happens that the cell tries to maintain the concentration of ATP at a higher level. A small change in the percentage of ATP results in a large percentage change in the concentration of other adenyl nucleotide. That means concentration of ADP and AMP. How? You have seen that ATP can be converted into ADP and AMP, and more of ADP can be further converted into AMP. That means they can be controlled depending on the amount of ATP. So whenever you look at this chart, it reflects the energy status of the cell. And a small change in ATP to large changes in AMP leads to the tighter control by increasing the range of sensitivity of phosphofructokinase. So look at how this ratio of ATP is to AMP, which reflects the energy charge of the cell, is going to control the enzyme phosphofructokinase. Now, so far we have talked about very simple mechanism. Now we are going to reach in the complexity of the reaction. So what happens? Phosphofructokinase is inhibited by citrate. And from where we get citrate? So far I have not talked about, so I'm just giving you a hint, and then we will discuss about the citrate in the next few lectures. And the citrate is the one which is being synthesized. As I said, pyruvate is formed by the glycolysis. Pyruvate is converted into acetyl-CoA. That part you have learned under the aerobic condition. And the acetyl-CoA, in presence of oxaloacetate, enters in a cycle called as a citric acid cycle or TCA cycle. And the citric acid is the first metabolite of this pathway, or this cycle rather. The citric acid is being synthesized, and because once you get the citric acid, it can produce a lot of energy. That means, what should it do? Should it activate phosphorylkinase or inhibit? When you have a lot of citrate, that means you have a lot of energy. So what it is doing is that phosphofructokinase is inhibited by citrate, because once citrate is synthesized, if citrate is accumulating, you will be, see, I'm not committing or doing anything. Once we talk about citric acid metabolism, you will learn more, because when you have more citrate, that means you have a lot of energy produced in citric acid cycle. So control is coming from citric acid cycle. So citrate or citric acid, which is the first compound of the citric acid cycle, it inhibits phosphofructokinase. And how it does is that it enhances the inhibitory effect of ATP. You will see, I'll show you the reaction and you will learn about that. So fructose, now there is one thing, a twist is coming over here. So far we were talking about fructose 1,6-bisphosphate. Can you recall? We have talked about fructose 1,6-bisphosphate, which is formed from the reaction of phosphofructokinase on a substrate, which is fructose 6-phosphate. So what fructose 6-phosphate does is it forms fructose 1,6-bisphosphate in presence of enzyme phosphofructokinase. Now I am naming another enzyme, another substrate, as well as product. Here substrate is same, but the product is different. What we are talking about is fructose 2,6-bisphosphate. So now this fructose 2,6-bisphosphate, I'll tell you in a few seconds, is one which is allosteric regulator of enzyme phosphofructokinase. So fructose 2,6-bisphosphate, it activates phosphofructokinase by increasing the affinity of fructose 6-phosphate and diminishing the inhibitory effect of ATP. So you have learned that how ATP does the job. Now you have another regulator. This regulator is nothing but fructose 2,6-bisphosphate. How it works? It activates phosphofructokinase. I'll show you in the enzymatic reaction. So what is a fructose 2,6-bisphosphate? It is given here. So what you have is, instead of at the first position, it's second position, which is getting phosphorylated. So it is the same kind of a molecule, but slightly different in terms of its chemistry. So instead of a 1,6-bisphosphate, instead of first carbon, which is going to be phosphorylated, here second one is phosphorylated. And this is not involved in the energy generation. It will not go further for metabolism. Its role is to regulate phosphofructokinase. Now, this phosphofructokinase is, as I said, is activator. It shifts the concentration. And what happens? So far, I have not talked about the enzymatic regulation, but when you see the allosteric enzyme, so you have phosphofructokinase and allosteric enzyme. So phosphofructokinase, this fructose 2,6-bisphosphate, it activates phosphofructokinase. How it does? It will bind with phosphofructokinase at allosteric site, and what it will do is that it will convert enzyme from T state to R state. What is the T state and R state? T state is referring to tense state and R state is a relaxed state. So during this process, once fructose 1,6-bisphosphate binds with phosphofructokinase, it relaxes the enzyme in terms of activity. This is I'm not talking about the enzyme structure, but yes, these are the two states. Refers to T state is where it is inactive, and R state or relaxed state is where it is active. This is the reaction which will show you that the reaction velocity, different concentration of fructose 6-phosphate, and it is showing how allosteric regulation occurs in presence of ATP. When ATP is high, that means initially the reaction doesn't occur. It's an allosteric regulation. So the enzyme requires high concentration of fructose 6-phosphate to become active. That means, what does it mean? Suppose I'm looking at the Km, here the Km will be somewhere here, right? And in this case, Km will be here. So by regulating the Km, ATP is controlling the reaction of phosphofructokinase. Is it clear to you? Hello?
Yes,
Yes, sir.
sir. It's clear.
Now looking at the activation of phosphofructokinase by 2,6-bisphosphate. Now fructose 2,6-bisphosphate is another regulator. Here ATP is one regulator, another regulator is where fructose 2,6-bisphosphate is regulating. So look at 2,6-bisphosphate, how it is controlling. Increasing the reaction, a different concentration of fructose 2,6-bisphosphate. Here you have this reaction. As soon as you increase the reaction of fructose 2,6-bisphosphate, it is going to be, regulated in a different fashion. So here we have shown how ATP and fructose 6-phosphate at different concentration can regulate these reactions. Now it's slightly complicated mechanism, and now time is over, so I would like to discuss in greater detail. But the point here to remember is that the enzyme regulates the concentration by phosphorating fructose 6-phosphate to fructose 2,6-bisphosphate. In a normal reaction glycolytic pathway, what will happen? That fructose 6-phosphate is converted into fructose 1,6-bisphosphate, but the some amount of fructose 6-phosphate can also be converted into fructose 2,6-bisphosphate, which will be controlling the reaction. So 1,6-bisphosphate is for the energy generation, and 2,6-bisphosphate is the one which is for the control of the reaction. And how this 2,6-bisphosphate is being formed? It is formed by another enzyme which is called as a phosphofructokinase. Conversion to 1,6-bisphosphate is by PFK1, that is means phosphofructokinase 1. Here, this conversion to fructose 2,6-bisphosphate is by enzyme phosphofructokinase 2. So these two are different enzymes as such, and they are controlling. One is controlling the synthesis of 1,6-bisphosphate, another is controlling the synthesis of 2,6-bisphosphate. 2,6-bisphosphate is the one which is allosteric regulator. 1,6-bisphosphate is the one which will go ahead for catalytic reaction. So here I will start from this part, how it is being regulated. So you will be actually looking at how the presence of high level of glucose can control the overall mechanism. So this enzyme, which has, this is a dual function enzyme. I will discuss more about it, because suppose I have a protein has got two different enzymes together. Their control can be regulated by a single trigger, and that trigger I will talk about. So I'm stopping at this point. Anything which is not clear, you want more emphasis, you let me know. I will take up in the next class. So if there are no more questions, I will stop at this point. I will log off, and thank you very much.
Thank you, sir.
Thank you, sir.